Hi, hello students. From today onwards, we are going to discuss about the cell biology. And for our better understanding, I divide the chapter cell biology into few parts. Okay. So first of all, these are the topics which covered in this video, in this particular video. The remaining topics is covered in next video. Okay. In this video, I'm going to discuss about cell biology, cytology differences, హిస్టారికల్ అకౌంట్ ఆఫ్ ద సెల్. అంటే ఒక సెల్ మనం ఇప్పుడు డిస్కస్ చేస్తున్నాం, సెల్ డివిజన్ ఎలా జరుగుతుంది, ఆర్ సెల్ మెంబ్రేన్ స్ట్రక్చర్ ఏంటి, డిఫరెంట్ థీరీస్ ఆర్ దేర్ టు డిస్క్రైబ్ ద సెల్. ఓకే, ఈ థీరీస్ అన్ని కనుగొనడానికన్నా ముందు దెర్ ఈస్ ఎ హిస్టరీ బిహైండ్ ద సెల్ స్ట్రక్చర్, బికాస్ సెల్ ఈస్ వెరీ వెరీ స్మాల్ స్ట్రక్చర్. మనం దాన్ని కంటితో చూడలేనంత చిన్నది. ఓకే, దాన్ని చూడడానికి అవసరమైన యాపరేటస్ లేని టైం నుంచి ఇప్పుడు దాంట్లో జరిగే ప్రతి ఈవెంట్ని మనం ఈజీగా ఐడెంటిఫై చేయగలిగే న్యూ ఆపరేటర్స్ వరకు జరిగిన హిస్టరీ ఏంటి అన్నది మనం డిస్కస్ చేస్తున్నాం. అండ్ వాట్ ఈజ్ ఎ సెల్, సెల్ అంటే ఏంటి అసలు అనేది మనం డిస్కస్ చేయబోతున్నాం. అండ్ ఆఫ్ కోర్స్ ద డిఫరెన్స్ బిట్వీన్ ప్రొకారియోటిక్ అండ్ యూకారియోటిక్ సెల్స్. And important cells means you already know that all cells are very important, but according for exam purpose or for our general knowledge purpose, some cells are there with a unique nature, like a smallest cell, largest cell, longest cell, like that. Next, prokaryotic cell structure also. Prokaryotic cell structure is nothing but a bacteria structure. So first of all, cell biology and cytology. So many persons use these two words as a synonyms. Of course, that is right. Cell biology is also called cytology, but that is not correct. It is not a complete correct answer. Okay. Cytology is the formal word, and cell biology is the general word. But cytology deals with what? Cytology. Cyto means cell. Cyto means what? Cell. Logos means study. Cytology is the study of cell, and it deals with what? Structure of cell, function of cell. That's it. Structure of cell means plasma membrane structure, internal cell organelle structure, and the function of the cells. That is described by the cytology. Cell biology deals with all the functions of cell organelles also, and it deals with hereditary information. It deals with chromosomes and cell division, chromosome structures, and metabolic activities which are conducted by or which are initiated by the hormone structures, that means chemicals. So biochemicals or biomolecules. So chemical aspects of the cell, physical aspects of the cell, cell division, chromosomes, that means hereditary, and of course cell structure, cell organelle structure, cell organelles functions, everything is covered in the cell biology. So we can easily say that cytology is the branch of cell biology. Cell biology is a great tree. In that great tree there is a small branch. That small branch name is cytology. Okay. Now we are going to discuss about the cell biology, not the cytology. Okay. Without any late, first we will start with the historical account of the cell. Try to remember few things which are very important. The remaining things try to just know only. Okay. 1590. Before 1590, there is no microscope. For the first time in 1590, F. Johnson and Z. Johnson, these two scientists discovered compound microscope. When compound microscope is initiated, they try to observe so many things, but it takes nearly 75 years to identify a cell. It takes how many years? 75 years. 1590 microscope is invented. 1665 for the first time Robert Hooke discovered the cell in cork plant. This is important. In cork slices, and he discovered dead cells, non-living cells. He discovered which type of cells? He discovered non-living cells, not the living cell. And he coined the term cell or cellula. Cellula or cell Cell means room, a chamber. He identified the chamber-like structures in the cork tree. They are actually non-living cells, dead cells. After then, it takes 10 years time to observe the living cells. Anthony van Leeuwenhoek. Anthony van Leeuwenhoek observed the living cells in 1675 after 10 years. Okay, exact 10 years. That is like bacteria he observed protozoa, he observed RBC, and etc. Some living cells are under the microscope. Later, it takes nearly 100 years gap for the living matter within the cell. Cells, okay. Within the cell, some living matter is absorbed by the Alfonso Corti and F. Fontana. Alfonso Corti and F. Fontana are identify the living matter within the cell cytoplasm. It takes 100 years time. And then next, within 10 years, within 9 years gap, F. Fontana discovered the nucleolus. We will discuss about the nucleolus also. It is a part within the nucleus. Nucleus is different, nucleolus is different, nucleoid is different. Three different words are there. Nucleus, nucleolus, nucleoid. Nucleoid is completely related to the prokaryotic cells. Nucleolus is a part of nucleus. Nucleus, you already know that. So we will discuss about these three words very detailedly in the next videos. He discovered the first time nucleolus in a skin cell. It is not a skill, sorry for the mistake. It is a skin cells of the eel. Okay. And then next, it nearly takes more than 70 years to identify the nucleus in Tradescantia. Tradescantia is the plant name, I think. In this plant, Robert Brown for the first time observed the nucleus. This is important. Leeuwenhoek, Robert Hooke, Robert Brown. Okay. And then next, after 4 years, Dujardin named the living matter present in the cell as sarcode. He gave the name as sarcode to the living matter within the cell. Next, Purkinje, J.E. Purkinje, after 4 years or after 5 years, he coined the term protoplasm. So this is the history behind the cell structure. In this, you have to remember a few scientist names and a few years. That is, 1665, Robert Hooke, who identified the non-living cell for the first time in cork plant. Next, in 1675, Leeuwenhoek, who identified the living cells, okay, different cells like RBC or something. Next, in 1831, Robert Brown, he observed the nucleus in orchid plants. Okay, so these three are very important. Next, what is cell? We use the word cell for several times, but what exactly cell means? What is the cell means? Cell means it is the structural and functional units of the any organism. Structural and functional unit of any organism, and it is also called basic unit of the life. Basic unit of life. You already know why it is called structural and functional unit of any organism, because the tissue is made by the fusion of similar types of cells. All cells together form the tissue, and tissues together form the organ. So it is the basic thing, and it perform different types of functions which performed by the organism. That means one organism can show the digestion, respiration, circulation, excretion. Same type of functions are showed by the cell also. Cell is also can digest. Cell also can show respiration. Cell also can show excretion. Within the cell, circulation takes place. Okay, it is also can product some types of hormones. So it is a structural and it is the functional units of the life. And it is also called a basic unit of life, because with cell is nothing but what? It is a protoplasm. Protoplasm means it is a fluid, like fluid it is. Within the protoplasm there is a controller. Controller is nothing but the DNA. Okay, controller this is. And under this controller there is some workers like mitochondria or Golgi complex or something. Everything is bound by a plasma membrane. This is cell simply. Okay, there is there is nothing less than the cell. ఇంతకన్నా చిన్నగా ఉండి ఇలాంటి వర్క్ చేసేది ఏదీ లేదు. ఇదే అన్నిటికన్నా చిన్నది. ఓకే, దిస్ ఈస్ ద బేసిక్ యూనిట్. సెల్ ఆర్గనల్స్ కెన్ నాట్ పర్ఫార్మ్ ఆల్ ద ఫంక్షన్స్ ఆఫ్ ద సెల్. ఒక సెల్ పర్ఫామ్ చేసే అన్ని ఫంక్షన్స్ని సెల్ ఆర్గనల్ పర్ఫామ్ చేయలేదు. But cell can do that. That's why it is the basic unit. It is the minimum basic unit we can consider as it is the life. Okay, we cannot consider the only nucleus as life. We cannot consider mitochondria as life, but we can consider a single cell as a life or living organism. We can call it as a unicellular organism, like amoeba, unicellular organism, paramecium, unicellular organism, and euglena, unicellular organism. But we never says that Golgi complex as a unicellular organism. So it is the basic unit of life, structural and functional unit of the life. It is the minimum thing. Okay, and we are going to discuss about different aspects of the cell. Clear? Right. Next, different types. First of all, mainly there is two types of cells are there. One is prokaryotic, another one is what? Eukaryotic cell. First, prokaryotic cell means what? Pro means first, beginning. Okay, we also use the same word pro for the games also. We do pro antam. That means he's the beginners. Okay, pro means beginner or at the starting time. Karyo means nucleus. Karyo means what? Nucleus. That means these are the cells before the formation of proper nucleus. That means there is no proper nucleus. Okay, DNA material is there, but it is not considered as the nucleus. U means true. Wherever you find the word U means true. Uri-helene or U metajoa. Okay, U coelom. U means true. Eukaryotic. Real nucleus is present here. Real nucleus means with nuclear envelope or nuclear membrane. If there is a nuclear membrane, we can consider as it is the eukaryotic organism. If there is no uh, nuclear membrane, that is a prokaryotic cell. That is the major difference between the prokaryotes and eukaryotes. Along with these, so many differences are there, not only the nuclear membrane. Okay, there is so many differences between the prokaryotic and eukaryotic. We will discuss one by one, prokaryotic and eukaryotic differences. Okay, so I'll write here the prokaryotic and eukaryotic differences. First prokaryotic and eukaryotic difference is what? Here it is the prokaryote and it is the eukaryote. Nuclear envelope is absent. Sorry, it is one more. Nuclear envelope is present. Envelope is present here. That is the first difference. The second difference, cell organelles also don't have any envelope. That means membrane bound cell organelles are absent. Membrane bound cell organelles. Membrane bound cell organelles like mitochondria is not there. Chloroplast is not there. Okay, endoplasmic reticulum is not here. So membrane bound organelles are absent, but membrane bound organelles are present. Membrane bound organelles like mitochondria. Okay, Golgi complex, endoplasmic reticulum. Okay, these are present. And of course, in both prokaryotic and eukaryotic, ribosomes are there because ribosomes are not membrane bound organelles. There is no membranes in the ribosomes. So ribosomes are present. But in prokaryote, there are 70s ribosomes. In eukaryotes, there are 80s ribosomes. They are what? 80s ribosomes. We will discuss about the ribosome structure. What is 70s? What is 80s later? Okay. And here there is DNA, but DNA is usually circle in form, and it is maximum. It is only n stage. Okay, it is not, there is no 2n stage. Maximum it is in n stage, and it is a round circle, and it is naked. Naked means it is not packed. It is not properly packed. But here it is in helical form. Okay, it is prepared as a chromatin thread. chromatin thread. It is packed with the histone proteins are there, and it is show two types of stages. That means n stage is there, 2n stage is also there. That means haploid and diploid, both stages it can perform in the eukaryotic cells. Not only this, the other difference is, yes, prokaryotes actually they are small in size, but they are, when compared with the prokaryotes, they are large in size. So because due to the lack of the cell membrane, just imagine as it is like this. Of course it is like this, maximum bacteria. So due to the lack of the nuclear membrane, just dotted line, just give a dotted line for our better understanding, but there is no cell membrane, nuclear membrane here. So it is present in the cytoplasm. So transcription, formation of mRNA, translation, formation of protein, everything is takes place in the cytoplasm. But here transcription takes place in the nucleus, and that mRNA comes out. Translation takes place in the cytoplasm. What is transcription? What is translation? We will discuss in the next videos. But there is two difference, two differences regarding to the transcription and translation. So these are the few differences between prokaryote and eukaryote. At the end of the cell biology, you can realize so many differences between the prokaryote and eukaryote. We will discuss later. Okay. Don't think as we are stopped here. It is just only brief introduction. After completion of the prokaryotic structure, we will move to the eukaryotic cell structure. Then you can realize the main differences. At that time also we will discuss so many differences between the prokaryote and eukaryotic cells. Now come to the prokaryotic cell structure, which is very very important. Okay. Prokaryote cell. Actually prokaryotic cells are nothing but bacteria. And blue green algae, cyanobacteria. We use the word cyno. Cyno means blue color in scientific language. For every color there is a word. Erythro, red color, erythrocyte. Cyno, blue color, blue algae, cyanobacteria. Okay. So chlorophyll, green. Chloro means green, like that, different names for different colors. So cyanobacteria, algae, blue green algae, and mycoplasma. Mycoplasma is the smallest cell. Mycoplasma is what? It is the smallest cell. Its length is 0.3 microns. Very very important bit. Mycoplasma is the smallest cell. Its length is 0.3 microns. Okay, smallest cell. If it is the smallest cell, what is the longest cell? Largest cell. Largest cell is ostrich egg. Ostrich egg is the largest cell. Okay, and there is a longest cell, neuron. Because of its axon length, neurons is the longest cells. Smallest, largest, longest cells. Smallest is the mycoplasma, that is 0.3 microns. Why mycoplasma considered as very very small? Because actually bacteria is nearly 1 to 3 microns, 1 to 3 microns. But mycoplasma is 0.3 microns, less than the 1 micron. Okay, so that's why it is the smallest cell ever. And mycoplasma we can consider it as pleuropneumonia like organisms, PPLO. Pleuro, where we use the word pleuro. Pleura is the membrane which is present around the lungs. This is pleura. Okay, and this mycoplasma is present in this pleura of animals also have the pleura around the lungs. In that pleura, scientists observe this organisms which are close to the mycoplasma or mycoplasma present in this mucus, in this flora. That's why they are called pleuropneumonia, because it causes pneumonia. Pleuropneumonia like organisms. Pleuropneumonia like organisms. Best example, mycoplasma. Mycoplasma is a type of pleuropneumonia like organism. Okay, and it is very very small in size. Of course, other prokaryotes are also there, and these two are not prokaryotes. Ostrich, egg, and neuron, these are eukaryotes. But for our revision purpose, ఇక్కడ మీకు స్మాలెస్ట్ సెల్ చెప్పాను కదా అని లార్జెస్ట్ లాంగెస్ట్ ఇక్కడ నేను మెన్షన్ చేశాను. బట్ దీస్ టూ ఆర్ యుకారియోట్స్. కమ్ టు ద ప్రొకారియోటిక్ సెల్స్. Prokaryotic cells are bacteria, blue-green algae, mycoplasma, and some actinomycetes. Other bacteria like what? Actinomycetes. Actinomycetes helps in agricultural field. It helps in the agricultural field. How it helps in agricultural field? రైజోఫోరా మొక్కలు ఉంటాయి కదా, లైక్ వేరుశనగ మొక్కలు. ఓకే, ఇట్ హెల్ప్స్ ఇన్ ద ప్రొటెక్షన్ ఆఫ్ ద టైప్ ఆఫ్ ద ప్లాంట్ రూట్స్. ఆ రూట్స్ని ప్రొటెక్ట్ చేస్తుంది. అండ్ ఇట్ యాక్ట్ లైక్ ఏ యాంటీ బ్యాక్టీరియం. ఇట్ ట్రై టు కిల్స్ ద అదర్ ఇన్సెక్ట్స్, or it has a high resistant power against the viruses. So it protects the plants. Actinomycete. It is also a prokaryote. Spirochetes are the long bacteria. Spirochetes are with 500 microns diameter. 500 microns diameter is there. 400 to 500 microns diameter. And Thiomargarita. Another thing, Thiomargarita. You can easily remember Margarita is a name of one person, very famous person, Margarita. Thiomargarita nambiasis. నంబిలాగా అంటూ ఉంటాం మనం. ఓకే, అది మీరు గుర్తుపెట్టుకోవచ్చు. థయోమార్గరేటా నంబియాసిస్. రీసెంట్లీ ఇట్ ఈస్ డిస్కవర్డ్ బై ద సైంటిస్ట్, అండ్ ఇట్ ఈస్ ద లార్జెస్ట్ బ్యాక్టీరియల్ సెల్. It has nearly 700 microns diameter. 700 microns diameter. Recently discovered. అంటే ఎంతకాలం ఇది నంబిలాగా ఉందన్నమాట. నంబియాసిస్, నంబిలా కూర్చుండిపోయింది. Thiomargareta nambiasis, but recently it is discovered and it is the largest one with diameter that is 700 microns. So all these are the prokaryotic cells. So I will erase this, then you can clear more. Okay, so these are the cells: bacteria, blue-green algae, mycoplasma, actinomycete, spirochetes, Thiomargareta nambiasis. These are the some prokaryotes, and we are going to deal with one of these prokaryote cell structure. Okay, we will take the bacteria structure, then that is we can apply to the remaining all cells, remaining all prokaryotic cells, bacteria structure if we discuss. Clear? Right. Now I'm going to discuss about the bacteria structure. So one prokaryotic cell has this different types of structures in its body. Okay, prokaryotic cell have this type of structures. First of all, the outermost cell wall. Bacteria or prokaryotes have the cell wall. Okay, outside of the cell wall there is a material that is called glycocalyx. So glycocalyx is also, I am drawing like this with this color, glycocalyx. I'll explain what is glycocalyx, what is a cell membrane, what is cell wall, everything. It is glycocalyx. So inside the cell wall there is a plasma membrane. There is what? Plasma membrane. Okay, and if it is movable bacteria, if it is moving, it has one flagella also. It has the flagella or many flagellas, depending upon the flagella number and their location also. Different types of prokaryotes are there. Flagella is there. and small hair-like structure, fimbriae is there, fimbriae, and inside the nucleoid is there, DNA material nucleoid, and cytoplasm. It is also called protoplasm. Okay, ribosomes are present. Ribosomes. Okay, and vacuoles are there. They are also considered as inclusion bodies. Inclusion bodies are vacuoles. Okay, and yes, mesosomes are there. Mesosomes. So we'll discuss every part one by one, one by one. First, we'll start with the envelopes. Envelopes means what are the which layers form the wall of the bacteria, wall of the prokaryotic cell. There is three layers. Outermost layer is called glycocalyx, and middle cell wall, and inner plasma membrane. Okay, before going to know about the prokaryotic cell structure, detailed structure, just revise once what we discussed up to now. We discussed about what is difference between cell biology and cytology. Cell biology deals with so many things, so many aspects of the cell. Cytology deals with only structure and function of the cell. Okay, and we discussed about the difference between prokaryotic and eukaryotic. Few differences. Prokaryotic means without nuclear membrane. Eukaryotic means with nuclear membrane. That is the major difference. The cell size, ribosome type, DNA transcription or translation, DNA structure, everything is secondary. And we remember the some years and scientist names. Robert Hooke, who discovered the for the first time non-living cell in cork plant. Next, Anthony van Leeuwenhoek, he observed the living cells like RBC or WRBC and some other cells. Okay, and then next Robert Brown. Robert Brown observed the nucleus. Okay, these are the some important scientists and their related years 1665 and 1675 and 1831. These are the years. Okay, now later we discuss the some few cells of the prokaryotic cells examples: bacteria and blue-green algae, actinomycetes. Okay, actinomycetes and the Thiomargarita. Thiomargarita is the recently discovered large bacterium, Thiomargarita numbiasis. Okay, and few largest, longest, and smallest. Smallest is the mycoplasma. Mycoplasma considered as pleuropneumonia-like organism. And RBC, I may forget. RBC is 7.0. The RBC size is 7.0 microns. Of course, 7 microns. You can say as the 7.0 or 7 microns RBC. And some other cells are also. Largest cell is the ostrich egg. Longest cell is the neuron. Now prokaryotic cell structure. This is the prokaryotic cell, bacteria. One by one we will discuss. First, the envelope. Focus on the envelope. I am going to discuss about these three layers. First layer is glycocalyx. What is the first layer? Glycocalyx. This is yellow color layer is called glycocalyx, outermost layer. So I'll write here outermost layer. That is the first point. And it is formed by the polysaccharides. Polysaccharides. And sometimes protein molecules are also there. And it is get from the outside, external. From the external side only it get that glycocalyx. And glycocalyx is in two different forms. If glycocalyx layer arranged loosely, the loose arrangement, that is called slime. Loose arrangement is called what? Slime. స్లైమ్ అంటే ఏంటి? మనకి అన్నం వార్చిన తర్వాత గంజి ఉంటుంది కదా, సెమీ సాలిడ్ లిక్విడ్ లైక్ సబ్స్టెన్స్. అది కూడా కాదు నిజానికి, అన్నం వార్చిన తర్వాత గంజి కూడా కాదు. గేమ్స్ ఆడుతూ ఉంటాం, మన పిల్లలు ఆడుతూ ఉంటారు గేమ్స్, దాంట్లో వేలు పెడితే మనకి పైకి కనిపిస్తూ ఉంటుంది, కొంచెం సాగుతూ ఉంటుంది లైక్ చపాతీ పిండి లాగా. that is called slime, loose arrangement of the polysaccharides. If they are arranged very tightly, that tight association is called capsule. It forms what? Capsule. Slime and capsule. Capsule is very difficult, very tough. It protects the inner layer, cell wall. It protects the cell wall and it protects the remaining plasma membrane also. And the main function of slime is to protect only, to protection. Capsule also can protect. Maximum the bacteria which have the capsule, that means glycocalyx, tightly associate with one another and it forms the capsule. They are present mostly in the positive, virulent, which causes damage, which causes disease. That type of bacteria have the capsule. ఏవైతే మనకి disease cause చేస్తాయో they have the capsule. Most of the non-virulent ఏవైతే disease cause చేయవో, ఏవైతే మన disease కలిగించవో, they have slime. So like that we can differentiate the glycocalyx. Glycocalyx is the outermost layer. I'll show the glycocalyx is slime and capsule difference. Okay, coating of molecules external to the cell wall made of sugars and proteins. They are made with what? Sugars and proteins. If they are loosely arranged, that is called slime layer. If they are highly organized and tightly attached, the tight attachment is formed the capsule. This is the example for capsule. This is the example for slime. Okay, this is outermost layer of the bacteria. Now we are going to the very interesting character of bacteria, very interesting character of prokaryote, that is cell wall. Cell wall is present in protozoans, that means prokaryotes, not protozoans. Prokaryotes have the cell wall, and fungus have the cell wall. Plant cells also have the cell wall, but cell wall is absent in the animal cell. Cell wall is absent in animal cell. Of course, we will discuss about the remaining cells also, but animal cells don't have the cell wall. But plants, fungi, and prokaryotes, all these are have the cell wall. Depending upon the based on the cell wall also, classification takes place. In that classification, they placed fungi under the kingdom Plantae at earlier stages. Because of the cell wall, presence of the cell wall only, fungus placed under the plants. But of course, later they will realize the difference between cell wall of the fungus and cell wall of the plants, cellulose and the pectin. We will discuss later about that. Now come to the cell wall of the prokaryote, cell wall of the bacterium. Okay, cell wall is outermost layer and it is present in most of the prokaryotes. This is made with the glycopeptides. This is made with what? Glycopeptides. And very important point, mycoplasma doesn't contain, mycoplasma don't have the cell wall. Mycoplasma don't have the cell wall. And it is very important bit. Mycoplasma don't have the cell wall even though it is a prokaryote. And that's why mycoplasma causes pneumonia. Pneumonia is very deadly dangerous disease if it is caused by the mycoplasma, because penicillium like antibacterial drugs never work on the mycoplasma. Actually, antibacterial drugs try to break the cell wall. It will die if there is no cell wall. Okay, that's why our penicillium try to break the cell wall. But here there is no cell wall in the mycoplasma. Okay, which is the smallest cell, which is very dangerous. Okay, cell wall is made with what? Peptidoglycan or murin. Cell wall is made with what? Peptidoglycan or murin. It is also called what? Murin. Okay, and based on the cell wall, the bacteria are divided into two types. They are positive bacteria and negative bacteria. Positive bacteria and negative bacteria are based on the cell wall. how they are differentiated into positive negative. Christian Graham created he prepared a strain to differentiate the bacteria based on the cell wall, based on the cell wall material. First of all, you have to understand the scenario. Cell wall is made with peptidoglycan. It is also called what? Murin. That means it is fully filled with two things. One is sugar, another one is protein. Both are forms the meshwork. This meshwork is called the murin or the peptidoglycan. Okay. Depending upon the peptidoglycan percentage or murin percentage, the scientist Christian Gram tried to differentiate the two types of bacteria. Okay, he prepared a strain that is called crystal violet. What is that? Crystal violet. Crystal violet అనే ఒక strain he prepared a crystal violet strain. Okay, ఇతని crystal violet strain ఏం చేశాడంటే he take the bacteria into one slide and he pour the crystal violet strain on it. Crystal violet strain he poured on the bacteria. So bacterial cell walls are receiving that crystal violet, they are turned into the purple color. They turn into purple color because bacteria cell walls absorb this crystal violet. Immediately they turn into the purple color. Okay. And of course later he added a mordant iodine. Mordant iodine. Why he used the mordant iodine? With the mordant iodine, when he pour mordant iodine in it, that mordant iodine helps in the attachment of crystal violet with the cell wall. Cell walls are closely attached with the crystal violet. Okay. Now, then he used the alcohol. He pour alcohol. He is very naughty. He is not satisfied with this experiment. He added alcohol. Actually, alcohol washes the crystal violet. Alcohol washes the crystal violet. So the bacteria which turned into the transparent, which loses their purple color, they cannot attach with the crystal violet. Okay, they are called negative because they are not closely attached with the crystal violet. They are called negative. But few bacteria, even though we are washing with the alcohol, we are washing with the water molecules, they never lose their color. They remain same in the color. They are considered as the positive. Why this happens? Why some bacteria never lose their color? Why some bacteria lose their color after washing with the alcohol? Because the bacteria which have more peptidoglycans, they are closely attached with the crystal violet, they turn into the purple. The bacteria which wall made with less peptidoglycan, they lose their color. And very important another point, teichoic acid. What is that name? Teichoic acid. Teichoic acid is present in positive bacteria. It is present in where? In positive bacteria. Because of the teichoic acid and more murin and teichoic acid, they never lose their color. Even though they washed by the alcohol, even though they washed with the water molecules, they never lose their color. They remain same in the purple color. But the bacteria which don't have the teichoic acid, teichoic acid, they are considered as negative because lack of the teichoic acid, they lose their color. Of course, peptidoglycan is also there, but very, very less in quantity. That is the Gram experiment. That is what? Gram experiment. So based on the cell wall, Gram conduct an experiment, and based on that, he confirmed, he divided the bacteria into two groups, Gram positive bacteria, Gram negative bacteria, like this. This is the experiment. Try to understand this experiment. First, he take the some bacteria into the slide. and he applied crystalline white, crystal violet, very important, crystal violet. After crystal violet, mordant iodine he used. When he used the mordant iodine helps in the attachment of crystal violet with the bacterium. Later he washed with the alcohol and water also. Once he alcohol, once we added the alcohol, the bacteria which don't have the teichoic acid, which have very less amount of glycol, that means peptidoglycans, they lose their color. They are the these. Okay. Later he used the word saffron. He used the saffron. We already know that. We use that saffron in our botany and zoology laboratory for not zoology, botany laboratory for the coloration. When he applied the saffron, the bacteria which loses their color, they turned into the red color. That means they don't have the teichoic acid. They are negative. And even though we use the saffron, they are still purplish in color because they have teichoic acid. They are attached with the crystal violet. They are gram positive bacteria. Clear about this gram positive or gram negative based on the cell wall. So we discussed about cell wall and glycocalyx. Glycocalyx is made with the carbohydrates and proteins. Two types, slime and capsule. Cell wall is made with teichoic acid and peptidoglycans. Next, plasma membrane. Plasma membrane is like this. The innermost layer is called plasma membrane. And plasma membrane at some positions is inverse and forms mesosomes. And forms what? Mesosomes. Of course it continues, but at some certain positions it enters into the cytoplasm and it comes back like this. These are called mesosomes. Sometimes it is tubule like structures like this, tubule like structures. Sometimes it is vesicle like structures. Sometimes it is lamella like structure, lamella like structure like this. So this is the plasma membrane. Maybe it is vesicle type, maybe it is a tubular type, or maybe it is the lamella type, so many layers. However, plasma membrane inverts into the cytoplasm and forms the vesicles. These are called what? Mesosomes. Mesosomes. Different types of mesosomes, like vesicle types of mesosomes, tubule type of uh, mesosomes, lamelliform, lamelliform mesosomes are there. Different types of mesosomes are present, are formed by the plasma membrane. So if it is the plasma membrane, the outer layer is cell wall. This is the cell wall. Cell wall is made by the what? Peptidoglycan. Okay, teichoic acid. Peptidoglycan and teichoic acid. If it is the cell wall, the outermost layer is what? Capsule, or it is the slime. That is nothing but glycocalyx. Glycocalyx made with what? It is prepared by the proteins and sugar molecules, carbohydrates and proteins. Okay, these are the three layers. Mesosomes. Mesosomes functions are, it helps in the DNA replication. It helps in the DNA distribution. When prokaryotic cell divide into two daughter cells, it guide where the cell should be divided. It guide from where to where the DNA is distributed. Some amount of DNA enter into this side, some amount of DNA enter into the B side, like that. Okay, and it helps in the formation of enzymatic activities. Okay, it increases the surface area, increase surface area for enzymatic content. Enzymatic content. So these are the few functions of the plasma membrane, mesosomes. These are the functions of what? Mesosomes. Mesosomes are the convolutions of the plasma membrane. Plasma membrane enter into the cytoplasm and it forms vesicle-like, tubule-like, lamella-type structures. These are helpful in DNA replication, DNA distribution, while the cell division, and increase the surface area for the enzymatic content. And for example, if it is the cyanobacterium, cyanobacterium means blue bacterium. If it is the cyanobacteria, within this mesosomes there is a chromatophores. There is chromatophore. Within the chromatophore, within the chromatophores, there is pigments. Within the chromatophore, pigments are there. Depending upon the bacteria, if it is the blue bacteria, cyanobacteria, these mesosomes helps in the photosynthesis activity because within the mesosomes uh, pigments are there, photopigments, which can help in the photosynthesis. This is one type of another function. Photopigments are there. And another function of mesosomes is mesosomes in some cells helps in the cell wall formation, helps in the cell wall formation. The outermost cell wall, it is formed by the mesosomes only. Mesosomes forms the cell wall. Like this, three different types of layers are there in the prokaryotic cell. Okay, just revise once. These are the mesosomes. While mesosomes develops, mesosomes divide. It is the mesosome actually. And while the cell division takes place, it guides where the cell division takes place, and it guides the nucleus. Okay, here chromatin thread attached with the this, and it is divided into two parts, and it guides the DNA into two daughter cells. This is the function of mesosomes. Okay, so outermost layer glycocalyx, it is formed by the proteins and carbohydrates, and it is a slime and capsule. The middle is a cell wall. Cell wall is made with peptidoglycans, murin, it is also called murin, and teichoic acid is also there if it is the gram positive bacteria. And we discussed how the gram positive negative bacteria are identified by the Christiana gram. And cell membrane, plasma membrane, which is involuted to form the which type of structures? Mesosomes. Next come to the flagella. Flagella, okay, we'll do one thing. We will stop up to the three layers. From the remaining structures, flagella, ribosomes, and internal structures, we will discuss in the next video. So prokaryotes and eukaryotes also will discuss in the next video. So thank you.